Hi everybody, I'm so incredibly sorry that lecture is going to have to be recorded for today. This is my backup plan. My husband unfortunately had a death in the family, and so we had to go back down to South Bend. And so if you're listening to this, it means I wasn't able to get back in time. On the bright side, we still get to have lecture. And on the downside, I couldn't do it live, and I do apologize. This will be the one time, barring anything else crazy happening, that we'll do this semester, because I much prefer live lectures. So today we're going to focus on adaptive radiation. We'll look at the Anole lizards. They're super fun. We'll also talk about the fossil record and radiometric dating. Okay, we'll talk about some of the bigger events in the history of life on Earth, things like mass extinctions, mammalian radiation, and we'll discuss the fact that really birds are tiny dinosaurs, which is actually kind of a cool thing. Still great in your exams. Going to be doing that for a while. I imagine you all can understand. So, all right, let's go ahead and get started. Now, when you have a speciation event, and if you'll recall, we've talked about this already as far as how it can happen and so forth. But if it happens really quickly, it's basically known as an adaptive radiation. Okay? You start out with a single ancestral species, and this rapidly diversifies into many descendant species. Remember, according to the biological species concept, what does it take to make a species? Okay? You've got to have some barrier to getting gene flow in between the two. Okay? The idea then is that the descendant species diverge into genetically different forms that occupy distinct habitats. This might bring about the question, how fast is rapid speciation? Well, it's actually a really good question. There's a really great example of this where the Anole lizards, I don't know if you've ever seen Anole lizards before. If you've ever been to Florida or any one of those places, these little guys run around everywhere. They're super cute and super wild, and they hate humans, and they try to hide as fast as they can. Well, there was a common ancestor of all the Caribbean Anole lizards, okay, and I think even people down in Florida call them Izzies. And then eventually they split up and grew, diversified, and ended up in Puerto Rico, ended up in Cuba, Hispaniola, Jamaica. But what's interesting is they split into these different areas, but within each of these areas they occupy different niches or different habitats. And so some will live on twigs, others in the canopy, and the next slide actually provides more information on this. Now, as I said, they ended up evolving relatively quickly. We know that different Anole species have different adaptations, and they will live in different parts of their habitat. Some live in the crown of the tree, those are the big guys. Others live on the trunk. Others live on the ground. I told you before, and I'll tell you again, I could never live in a tree because I'm too ungraceful and I would fall out and die instantly. But what's interesting about this from a phylogenetic perspective is that these guys speciated so quickly that it's almost impossible to determine what came first. In fact, if you look at it on a tree, it's represented by a polytomy. You remember that? That's when they all branch out from the same common ancestor, and you can't really figure out who's from who. Okay? So this happened relatively fast. We do know that. So here's a really good application question for you. So which of these 4 islands do you think acquired anoles most recently? Okay, so Cuba, Hispaniola, Jamaica, or Puerto Rico? Well, the way this is meant to represent, first off, pause this for a second and then take a shot at it. Okay? Now, if you've paused it and you got your guess, the next question is which one? Well, so what this is meant to represent is the y-axis represents time. And I know it's not exactly the typical y-axis that we think of, but going down to the common ancestor to the squares at the end, that represents time. And so what you want to look for is which one of the islands most recently accumulated the anoles, meaning which one had the time that was closest to those squares at the top. Okay? And unfortunately, if we were in class, I could explain this better. However, if you look at it, then J, or Jamaica, is the one where it's most recently have accumulated them because they didn't appear there until the branches there are there, in which case that's the closest to the squares at the top. Hopefully that makes sense. So the answer is C, because again, the y-axis represents time, and the x-axis kind of represents space, geographic space, and populations. And so Jamaica, because it was the one that came about the most recently, because the oldest is at the bottom and the newest is towards the top, that's why the answer to this is C. Oh, look, more practice. Okay, so take a moment. Dewlap, okay, is used by, it's this colorful skin flap. and it's used by males to attract females. What type of selection is this? Hopefully you guys said sexual selection, okay? But this is really important for finding mates of the same species because females of the same species will know to look for these colors. Given this information, what type of reproductive isolating mechanism is dewlap color? Take a moment, take a guess, pause lecture, and when you're ready, start again. Now at this point, it might look either A or B could potentially be answers, and you would be correct, okay, due to the dewlaps. However, the one thing I forgot to tell you is that, well, not forget, but challenge to tell you, is the fact that they use these dewlaps in communication, okay? So if they're using these to communicate, which is what they do, and this is how they communicate with females, it's going to be behavioral isolation, okay? Hopefully that makes sense. And no, I wouldn't forget to tell you something on the exam. So as you all know, I try to do my best to be fair and forthcoming. When it comes to the different habitats, these little anole lizards definitely are adapted for each. Trunk lizards tend to have long legs and they're really agile and they can quickly run up and down tree trunks. Twig lizards, though, have shorter legs, they live on the small twigs and kind of keep to themselves. So, given this, what do you think is the reproductive isolating mechanism if you consider this? Give it a moment, take your guess, pause lecture, and when you're ready, start again. So in this situation, hopefully you guys chose C, because they're living in different ecological niches, therefore it's going to be ecological isolation. And if that doesn't make sense, please let me know. Once again, we have a nice application question, and again, I'm gearing you up for 172 or for your advanced bio courses that you're going to take. So take a moment, read the question, which of the following is most likely explanation for how lizards introduced to a new island without trees acquired shorter legs. Choose among your answers, pause lecture, and once you're ready, start again. Now remember, in order to pick the right answer, you can eliminate the other ones. Okay, so in order to choose it, it's got to be 100% correct and make sense. So let's eliminate A. A new recessive mutation occurred, causing shorter legs, which is inherited by all individuals in the next generation. Well, good luck with that. Okay, not going to happen. B. The offspring of the original parents developed shorter legs from climbing in bushes. That is known as Lamarckian evolution and does not happen. C. Some of the offspring of the lizards that were introduced to the island had shorter legs and were more likely to reproduce than their siblings. Aha! That one sounds pretty good. Sounds like differential reproduction to me. And last but not least, hybridization between different lizard species produced lizards with shorter legs. This was never said. Okay, so nuke that one. That leaves us with C. So if you're looking at the slide, you're wondering, well, what is rapid speciation and associated with? And I would say, aha, that is an excellent question. The answer happens to be ecological opportunities and or morphological innovations. A great example of this would be the Hawaiian honeycreeper. Okay, so the Hawaiian honeycreeper started out on just one of the islands, of course the oldest one, and over time spread to the other islands. And as they did, there were whole new niches to occupy and space to explore. And so once the plants were actually going to these other areas, they could specialize and adapt and try to make the most of their new environments. So rapid speciation can happen, relatively quickly. In fruit flies, it can happen certainly very fast. We've talked about that before. It just depends on how old the organism lives and so forth and what their genetics are like. So this most likely happened right after the islands were formed and cooled, and then the plants could move in and start settling and then start adapting to their new environments. Now, when it comes to phylogenies and the fossil record, these tend to be the tools that biologists use to study the history of life. For the rest of this lecture, we're kind of going to be focusing on the history of life. In some ways, this kind of kills me because I have an entire semester-long class that focuses on this topic, and I'm supposed to cover this in half a lecture. Yeah, right. I'll do my best. We're going to hit the cliff notes of the cliff notes of the cliff notes, okay? However, there are some interesting things to talk about. First off, if you've ever heard of the White Cliffs of Dover, these are primarily composed of fossilized phytoplankton, otherwise known as forams. And so many of the structures we see today in the geologic record were formed by living critters long ago, which is actually pretty cool. Now, I have always been a huge fossil geek, and my husband will tell you as we go from rock show to rock show and all sorts of cool things. I have lots of fossils that I've collected over the years, and we are never moving again because as we moved into our current house, we were cart loading boxes of fossils, and I was getting all sorts of looks, dirty looks from my husband as he's looking at the rocks thinking, why do we have to move these rocks? But fossils are cool, okay? And they're actually quite rare because in order for a critter to be preserved into a fossil, the process has to happen just right. So, one example of this, the fish dies, sinks to the bottom of the lake. Okay, so then the fish rots and only the bones are left. This is typically what happens. The fish then gets covered with mud, okay, and millions of years pass, the mud turns to rock. Over time, the bone matter is completely changed into minerals, and the fish is now a fossil. Now, if you read your book, read the book. There are two main types of fossils. What might they be? If you did reading ahead of time, you can fill this out, and it's a piece of cake. So stop lecture here, and go ahead and take a shot. Now, if all went well, okay, you know that body fossils are basically direct evidence of a living critter. So we're talking bones, shells. We actually have tissue preserved in ice. Mammoths, woolly mammoths are pretty famous for that. Amber does manage to keep insects and flowers well preserved, although it does not preserve the DNA, unlike what Jurassic Park tells you and gives me a headache every single time I see the movie. So I used to work in the field of ancient DNA long ago. Amber is a horrible preservative for DNA because it's very porous, and usually when people do extract things from amber, it's just bacterial contaminants. Anyway, that's neither here nor there. So leaves can also be preserved as direct evidence, petrified wood, and so forth. Now, trace fossils are basically indirect evidence of a living critter. So tracks, holes in leaves, burrows, chemical remains, something along those lines. And the reality is when you're dealing with a fossil record, you got to take what you can get because you certainly can't be super picky because there's not a whole lot out there anyway. There are so many limitations of the fossil record. This is just a beginning list, okay? Habitat bias. A lot of critters that die on land are not necessarily well preserved because they get scavenged, whereas those that are in the ocean might get buried by sediment relatively quickly, and therefore they get preserved. And so, ocean critters might be preserved better than land critters. Taxonomic bias for the same reason. If you have critters that are sedentary and they don't move around much, most likely to be preserved, whereas others that move around a lot, probably not. Temporal bias. Certain areas in time are better preserved than others, as well as abundance bias. Okay, certain fossils are more abundant than others. Realistically speaking, you also have to consider the fact that we haven't even found a fraction of the fossils that are out there, and that only represents a fraction of life that's ever lived on Earth. Okay, so very, very few fossils of the critters exist, and very few critters were ever turned into fossils, so if this tells you anything about how the fossil record is lacking. You might also be asking yourself, how in the world do they determine how old fossils are? And I would say, that is an excellent question. So what scientists use is called radioactive decay, geological dating, radiometric dating. All those terms basically mean the same thing. So what happens is in the world, there are what are called isotopes, and these are basically the same element, but they have a different number of neutrons. Some of them are stable, some of them are not. Carbon-12 is stable, carbon-14 is not. What happens is you will have an unstable radioisotope decay at a constant rate into the decay product, okay? Scientists know how long it takes for this to happen, and that also allows them to determine how they can use this for dating. Now let's go through this table as an example. Carbon-14, okay, is unstable. And so what happens is scientists will take a chunk of material that they want to get a date on. They measure how much carbon-14 is present, and then they also measure how much nitrogen-14 is present. Now what's really important to know is that carbon-14 does not decay into carbon-13 or carbon-12. Carbon-14, when it decays, it turns into nitrogen-14. Okay, that's just the basic chemistry laws. Now, a half-life is how long it takes for half of the material to decay. Okay, so the half-life of this is 5,730 years. This means you can use it to date fossils that are anywhere from 100 years old to 30,000 years old. Okay, the way you do this, again, is you measure the amount of carbon-14 and you measure the amount of nitrogen-14 that's present, and then allows you to determine if your fossil is young or old. For example, let's say you have your fossil and you find out you have a bunch of carbon-14 and not much nitrogen-14. Okay, so think about it. A whole bunch of carbon-14 that is not yet decayed into nitrogen. Is your fossil very old or very young? Should be very young, okay, because it's not had a lot of time for the carbon to decay into nitrogen. Now, let's say you have another fossil and you measure the carbon and nitrogen in it and you find out there's a bunch of nitrogen-14 but not a lot of carbon. So is your fossil young or old? In that case, it's going to be old because a lot of the carbon has decayed into nitrogen. Okay, and the same works for potassium turning into argon, rubidium turning into strontium, and so forth. These all decay at a different rate, and that's really important because you use different elements depending upon how old you think your fossil is going to be. If you're somewhere between 100,000 to 4.5 billion years, use potassium-40 and argon-40. Uranium, okay, decays into lead, and potentially the half-life is 710 million years. You guys get the idea. Okay, hopefully this makes sense, and now let's go ahead and work through a problem to apply this. Now, using the previous table in the previous slide, so make sure you download your PowerPoint slides because you're going to need to take notes. Okay, what I want you to do is solve this problem. So pause lecture, read the question, okay, and you can use the previous table if you need to, and then I want you to figure out how old your sample is. And I want you guys to be able to do this because you're going to see something like this on the next exam. Okay, so here we've got uranium-235 to lead-207, half-life of 710 million years. Okay, you find 25% uranium and 75% lead. Based on this, how old is the sample? Hopefully you've chosen D. Now the reason we know it's D, okay, is because we said previously, and they said above, the half-life is 710 million years. So at 710 million years, you'll have 50% uranium and 50% lead. But that's not what we have. We have 25 and 75. So that would happen after going through another half-life, okay, which you go through a half-life again, and that leaves you with 25% uranium and 75% lead, okay? That means that the fossil is 1,420 million years old. So let's walk through this again. You know that at 50-50, okay, 50% uranium, 50% lead, one half-life is going to be 710 million years. However, that got split again. And so it went through two half-lives. That's why you've got 25% of the parent, uranium, 75% of the daughter product, as it's called, otherwise known as the lead. Okay, this is what it turns into. This is stable. Therefore, it's gone through two half-lives. So you take 710 times 2, and that gives you 1,420 million years, and that's how old the fossil is. So let's try this again. After 3.9 billion years, the ratio of potassium-40 to argon-40 in a rock would be what? Go check out the half-life. Figure out how many would have to go by to get to be 3.9 billion years, and that's going to give you your answer. Okay, so one half-life for the potassium to argon-40 is going to be 1.3 billion years. 3.9 billion years means it's gone through three half-lives. Take 1.3, multiply it by 3, that means it's gone through three half-lives. Okay, so remember the first half-life is 50-50. Second half-life is going to be 25 and 75. The next half-life, the third half-life, is going to give us 12.5 of the parent unstable potassium-40 and 87.5% of the argon-40 stable daughter product. Okay, so the parent product is what's going to do the decaying, by the way, and the daughter product is what it gets decayed into. And hopefully this makes sense because you've gone through three different half-lives here. Oh, look, a nice little table summarizing this. Okay, and as you can see, the blue is the decay of the unstable parent radioisotope. The red is the buildup of the new decay product, otherwise known as the daughter product, which is stable. Number of half-lives, one half-life it's 50-50, two half-lives it's 25-75, three half-lives, and so on. Okay? Make sure that this makes sense because I'm going to ask you something like this on the exam, and if it doesn't, please let me know and let's chat. Okay, now we have to somehow cover 4.5 billion years of life on Earth in about, oh, I don't know, half an hour. Bear with me. Okay. The time Earth has been in existence is somewhere between 4.5 to 4.6 billion years ago. Okay, now initially there wasn't a whole lot going on as far as life, and life has been around a while, but to be perfectly honest with you, most of the time of Earth's existence, it was actually single-celled and kind of invisible. All right. Now, in order for scientists to talk to one another, they've divided Earth's time up into several different periods and eons and eras, and hopefully this sounds familiar. The Precambrian time basically encompasses everything from the formation of the Earth around 4.6 billion years ago, chugging through the different eons, the Hadean, Archean, and Proterozoic. And I'm actually going to cover these in a little bit more detail in the next few slides. But what I want you to pay attention to here is that certain fossils, okay, the first evidence of life can be one of the most challenging things to discover, as well as when photosynthesis began, okay. Then one of the next big events was the first evidence of an oxygen-rich environment. And now I always say, if you ever invent a time machine, be careful how far back you go. Because if you go past too far, there won't be any oxygen in the atmosphere, and there won't be any going back. Just kidding. Anyway. Then we had about the first eukaryotic microfossils of 1.8 billion years ago. The first animal fossils, by the way, didn't come around until about 600 million years ago. Once again, we're fashionably late. Now, 4.5, 4.6, give or take 0.5 billion years, Earth was formed during the time period known as the Hadean Eon. The Earth was getting slammed by meteors. Lots of volcanoes, no life, no oxygen. Eventually, the Earth's crust would cool, okay, and liquid water would finally start to condense here. But it would take a while for this to happen, and so Earth actually had a pretty violent beginning. The next time period is known as the Archean Eon, so 4 to 2.5 billion years ago. We have our oceans, still lots of volcanoes, no oxygen. The first prokaryotes would come about, though, and then eventually photosynthesis would come about. And this actually would be really important because scientists hypothesize that it's thanks to photosynthesis that we have oxygen in the atmosphere. Gotta tell you, I'm a pretty big fan of that. Now, the Proterozoic Eon was between 2.5 billion years to 542 million years ago. So stromatolites would become very common, okay, and stromatolites still exist today, if you guys are curious. They're little ecosystems of single-celled critters, and they're actually pretty cool. The atmosphere and the oxygen, goodness, the atmosphere has been oxygenated, and the ocean is as well. That happened around 2.4 billion years ago. This was actually called an oxygen catastrophe because any critter that could not live in the presence of oxygen went through a mass extinction event. The first eukaryotes would come about, at least our eukaryotic cells, around 1.8 billion years, and then eventually, as I said, the first animals, which came in the form of some sponge-like critters, was around 600 million years ago. Now, I would like you guys to pay attention to this slide and even zoom in on it if you can. You should be able to do that and kind of just get an idea of the brief, did I mention brief Cliff Notes version of the Cliff Notes of the history of life and different events as far as when they happened. Now, approximately 542 million years ago, we had what was known as the Cambrian explosion. And essentially what happened is conditions were right, and most animal phyla started to appear in the marine fossil record. So many of the critters that we know and love today, their ancestors started to appear way back at this time. And so it happened so quickly that it's called an explosion. Despite this name, it was actually more of a gradual event. Animals increased significantly in size and diversity, and it took about 60 million years, okay, in order for us to have a lot of complexity the way we would expect the fossil record to be. However, you know, it depends on how you look at it. 60 million years, many different critters evolved. Still kind of could be an explosion. In fact, one could view this explosion as an adaptive radiation of the different animal groups, aha, we've heard that before, that may have evolved earlier, sometime before 542 million years. Now, this critter is one of my favorites, I'm not going to lie. It's called a hallucigenia, and you can kind of see why. It was named right around the time long ago when one could imagine hallucinating, such a critter, but according to the fossil record, these guys existed, and boy, they're interesting. Now, the earliest life, by the way, was all in the ocean. Okay, so, and we know that all animals are multicellular. We don't have cell walls. They have a gastrula. They have collagen proteins. Okay, we also know that choanoflagellates are the link to the first multicelled critters, which of course then would come about the sponges, and then from that true tissues would evolve, and then we would have our cnidarians, and then eventually our bilaterans, which just means that if you split those critters down the middle, you can only do it once in order to have mirror images, and humans are a really good example of this. So, but these guys eventually would evolve, and they are sort of the precursors to the critters that we know and love today. Now, keeping in mind that once again, we're only hitting the highlights of the highlights, we know that the Cambrian explosion was around 540 million years ago. Eventually, we would saunder onto land with fungi and plants. Would saunder first about 475 million years ago, then the arthropods about 420, and finally vertebrates about 365. Okay, we also know some of the bigger events, the KT extinction, that's when the dinosaurs finally kicked the bucket. That was around 65 million years ago, and then, 200,000 years ago, finally we made our very, very fashionably late entrance onto the scene. Now, we also know, according to the fossil record, that there are 5 mass extinction events during the time period Earth's existence. Okay, so a mass extinction is when you have some sort of catastrophic planet-wide environmental event. 50% or more of your species have to go extinct. Okay, when this happens, you can get ecological opportunities opened up. So generally, these tend to be followed by rapid adaptive radiation. Now, this compares to what's called a background extinction rate, which is basically just the average rate of extinctions over all of Earth's history. Okay, and what this is meant to show here is approximately when these 1, 2, 3, 4, 5 mass extinctions took place. Okay, the percentage of families that went extinct, and then going from 542 million years ago to the present when they happened. Now, each one of these had all sorts of interesting critters that existed at the time, and when the extinctions happened, of course, they ceased to exist. The most famous is known as the End Cretaceous extinction, otherwise known as the KT extinction, and this again is when the dinosaurs finally bit the dust. Now, this is most likely hypothesized caused by an asteroid impact, although I've talked to many scientists who feel that this may not have been the case. However, they do know that 75% of all species went extinct. All of the non-avian dinosaurs went extinct. Okay? There was a crater that scientists believe resulted from the impact of this particular asteroid. It was found in the early 90s in the Gulf of Mexico. And what's interesting about this is right around the 65 million year ago mark, there's a layer of sediment that is all around the Earth that contains what's called iridium. And iridium is generally only found in asteroids, and it's very rare on Earth. So those lines of evidence suggest that the asteroids really are what caused the dinosaurs to go extinct. Now, I don't know about all of you, but when I think of an asteroid impact, I think, oh, it's going to be really cool because you can still see the rock that came from outer space. But actually, that's not what happens at all. And I was kind of bummed when I saw my first asteroid crater out in Arizona, because basically what happens is everything is destroyed. Okay, and so the crater that is landed, scientists think landed about 65.5 million years ago, is shown here on a map in the Gulf of Mexico. And what happened is it obliterated the entire asteroid and left a huge crater because of the force of the impact. Once the asteroid did hit, it caused what was known as an impact winter, where there was so much dust in the atmosphere that it blocked the sunlight. There was approximately no photosynthesis for a year. Going to be kind of sad, okay? Temperature declined, and essentially there was a sulfuric acid rain. Again, time machine, be careful. Now, we know that the KT extinction removed many species, okay? However, it left a lot of real estate open for other species to adapt and radiate. Birds, mammals, lizards, snakes, ants, butterflies, all sorts of creatures just took off, whereas before they'd been hunkering and laying kind of low, trying to avoid being eaten by the dinosaurs. Now, I've said it before and I'll say it again, birds are the last living lineage from dinosaurs, okay? And you might be asking yourself, how do we know? Well, there's several different lines of evidence. One of the most important is known as an open acetabulum. This is basically an open hip joint, and this is what allowed the dinosaurs, certain dinosaurs, all of them actually. In order to be a dinosaur, you had to have this, okay, so you could walk on the two back legs. Now, some species of dinosaurs got so large that they eventually had to go down to walking on four legs again, okay, but the living birds kept that open hip bone and obviously walk on their back legs, and then their arms were modified to turn into wings. So that's just one line of evidence that the open hip bone, that birds are related to dinosaurs. Now, the type of dinosaurs birds are related to are known as theropods, and this is because they share particular traits with the theropods. They have a wishbone, they have what's called a semilunate carpal, they have hollow pneumatic bones, which of course the purpose is to make them much lighter. Okay, so this is just a line of evidence. If you compare a bird fossil with an Archaeopteryx skeleton, I don't know if you guys have ever read the cartoon BC, but they always have the Archaeopteryx. Those are one of my favorite little critters that they draw. Okay, but both of these can look very similar, and you can see a lot of similarities. Keep in mind, we don't have DNA from dinosaurs, despite what Hollywood tells us, okay? And so we have to go based on the fossil evidence alone, but you can see a lot of similarities between the Archaeopteryx and the pigeon, okay, that it's reasonable to assume that they're related because of all these similarities. Now, I'm going to admit, ethically, I'm kind of torn with what I'm going to describe to you next. The scientist in me thinks this is so cool, and the human in me, well, it's still really cool, as long as the animals are okay. There was a study conducted not too long ago where proteins that are normally produced on a chicken's face were blocked. What ended up happening is the chickens failed to make beaks, and instead they made snouts that are similar to the ancestral state, otherwise known as alligator-like. How cool is that? Okay? Just by blocking these proteins, their snout changed and started to look like some of those ancestors from long ago. In this same experiment, once the proteins were blocked, the bones changed, okay, and the palate changed as well. So instead of having a beak, the palate, which that's, the upper roof of our mouth, also changed as well, okay? So a single gene caused a change in many traits. This is known as pleiotropy. Not polygenic, this is pleiotropy, okay? And seriously, how cool is that? One gene to actually see all these differences and basically take chickens back closer to their relatives. That's really awesome. Now, additional information came about in 2016 when a 130 million year old bird fossil was discovered. This guy had a beak, no teeth, and feathers, okay, and also had reptilian type keratin. Once again, sort of a link between dinosaurs and birds from a morphological perspective. Now, there are three major lineages of mammals, okay, so monotremes, marsupials, and placentals. So the monotremes are located above, okay, then we got our marsupials, kangaroos, then we have our placentals. So which group of mammals are most closely related to primates? We've said it before, placentals, all right? Okay, hopefully this lecture will upload okay, and I will also plan on including a couple of videos for you guys to watch. I will talk to you live on Friday, and I will still be grading exams, and I'm sorry, I'm doing the best I can. I wish you a wonderful evening, and I will chat with you very soon.